Alright, so we'll be looking at carbohydrate metabolism in the brain. Carbohydrate metabolism in the brain. Now we know that we have 4 macromolecules, right?
Yes.
The 4 macromolecules are carbohydrates, proteins, lipids, and what? And nucleic acid. Good. Now these things can also be metabolized in the brain. So let's look at one of them, carbohydrates metabolism in the brain. Now, the storage form of carbohydrates in animals is what? Glycogen, right?
Yes.
Now, have you ever asked yourself, how does your brain get energy? Because if, okay, you know, you read when they say someone is mentally stressed or you are mentally tired, how does your brain even get energy? How does your brain get energy? Have you ever asked yourself some questions? Now, after taking carbohydrate-rich food, the carbohydrates, now look at this. Okay, let's assume this to be your intestine, because it's obviously not mine. Now, after taking food, the food passes through your bullet or esophagus to your where? Your stomach, right? Yeah. From your stomach to your small intestine, and digestion ends in the small intestine, right? The nutrients, which is monosaccharide and glucose, is absorbed into the bloodstream. Now, this glucose can also be found in the brain, because the brain depends on glucose as energy source. Glucose is carbohydrate, is it not? Yes. Now, the brain depends on glucose as energy source. So how does the brain utilize glucose? Now, once glucose gets into the cell, we have glucose transporters. Once it gets into the blood, we have glucose transporters. We call them GLU, glucose transporters, that help to transport the glucose to different parts of the body, different cells of the body. Now, specifically, GLUT1, because we have up to GLUT5, that's GLUT1, GLUT2, GLUT3, like that. GLUT1 transports glucose to the brain cells. Now, when the glucose gets to the brain, what happens to the glucose? The glucose can undergo glycolysis. Glycolysis is the breakdown of what? Glucose. So the glucose can be broken down. The glucose can be broken down into what? Pyruvate. Remember, in glycolysis, glucose is first converted to what? Glucose 6-phosphate. From glucose 6-phosphate to what? Glucose 6-phosphate. From glucose 6-phosphate to glucose 1,6-bisphosphate. Glucose 1,6-bisphosphate is then cleaved to dihydroxyacetylphosphate and what? Glycolysis. Dihydroxyacetylphosphate cannot continue in the pathway, so it will be converted to another molecule of glycolysis. Now, glycolysis is converted to, I'm looking at your mouse. Glycolysis is converted to what? 1,3-bisphosphoglycerate is converted to what? 3-phosphoglycerate. 3-phosphoglycerate is isomerized to what? 2-phosphoglycerate is converted to what? Phosphoenolpyruvate. And phosphoenolpyruvate is converted to pyruvate. Now, in this course, we are not interested in those long pathways. What we are interested in is, we started from glucose and we are ending with what? Pyruvate. Now, the glucose in the brain undergoes glycolysis, breakdown. So the glucose is broken down to release what? Pyruvate. Pyruvate. Good. Now, what happens to the pyruvate? Now, the glycolysis takes place in the cytoplasm. That's where glycolysis takes place. Now, this pyruvate here, let me write it out. These are glucose to pyruvate now. I'll say this process is called what? Glycolysis, right? Now, this pyruvate is transported from the cytoplasm into the mitochondria, right? It is in the mitochondria that the TCA cycle takes place. Are we getting it? It is in the mitochondria that the TCA cycle takes place. Now, but before entering into the TCA cycle, pyruvate is first converted to what? Acetyl what? Acetyl. And we said the process whereby pyruvate is converted to acetyl is called what? Oxidative what? Oxidative decarboxylation. Oxidative decarboxylation. In the process, NADH is produced. NADH is produced. Why is it called oxidative decarboxylation? Now look at the structure of pyruvate. This pyruvic acid. Now look at the structure of acetyl-CoA now. Check the difference. We just remove this carboxylic acid. That's why it's called decarboxylation. Oxidative decarboxylation. Redox is a redox reaction actually. Oxidation reaction is taking place. Now we remove this carboxylic acid group and we attach coenzyme A with the sulfide bond. That's acetyl-CoA. So pyruvate to acetyl-CoA. Now what happens to the acetyl-CoA? We'll come back to what is NADH. What happens to the acetyl-CoA? Remember, all these are in the mitochondria now, right? Yeah. Acetyl-CoA now can now go into the TCA cycle. TCA is tricarboxylic acid cycle. Tricarboxylic acid cycle. If you don't call it TCA cycle, you can call it what? Krebs cycle, right? Named after the person that first brought out the pathway. So and Krebs cycle. If you don't call it Krebs cycle, you call it what? Citric acid cycle. Now it is called tricarboxylic acid cycle because citric acid is tricarboxylic, like have 3 carboxylic acid groups. We are not interested in that. Now, the acetyl-CoA goes into the TCA cycle. And the purpose of TCA cycle is to generate more energy, right? Yes. Remember all these things we are breaking down glucose, doing this and that. We want to give energy to the brain. Yeah. Do we get that? Yeah. And the energy currency of the cell is what? ATP, right? Now, but we have energy-rich substances here, energy-rich. Now, you can recall with me that to enter the TCA cycle, acetyl-CoA combines with what? Oxaloacetate, right? Yeah. Oxaloacetate to form what? Citrate, right? From citrate to isocitrate. From isocitrate to what? Alpha ketoglutarate. From alpha ketoglutarate to what? Succinate. From succinate to what? Succinate to what? Fumarate to what? And manage back to oxaloacetate. But the good thing here is that, hello, the good thing is here is that in this course, you are not prepared to draw the pathway. Our main interest is in this pathway, NADH was generated, FADH2 by generated also. NADH was generated, FADH2 was generated also. Now, we generated NADH in glycolysis. Step 6 precisely, we don't want to go into that. And the link reaction, this pyruvate to acetyl-CoA is a link reaction between glycolysis and TCA cycle. Now, in this link reaction, we also generated NADH, right? Yes. Is it not? Now, in TCA cycle, we generated more NADH and more FADH2, right? Is it not? Now, what we are interested in is the NADH and the FADH2 generated. They are energy rich, but the body does not recognize them as energy source. What is the energy currency of the cell? ATP, right? So now this NADH and FADH2, we need to convert them to ATP. From our previous knowledge, we know that one NADH is equivalent to how many ATP? 3 ATPs, right? One FADH is equivalent to how many ATP? 2 ATP. Good. But how is it equivalent to ATP? How does it produce ATP? Now, this NADH and FADH2 will pass through the electron transport chain. It's called ET, electron transport chain. And as they pass through the electron transport chain, ATP will be produced. Electron transport chain is a very broad aspect, but let me just summarize it. Electron transport chain have 4 complexes. Hey, and for long
No, it's your turn laugh. It's incidental laugh.
Alright, so let's look at the electron transport chain. You see, this is not how it looks like, but just an illustration. Maybe as we go on, we'll learn the electron transport chain as a topic on its own. But now let's just get the idea of how NADH and FADH is converted to ATP. Just an overview. Now we have complex 1, complex 2, complex 3, complex 4. NADH can only bind to complex 1. So NADH can bind here. As NADH binds here, it will be converted to NAD+. Is that